Dear students, today we will discuss on diseases of brinjal and bhindi and their management. Among the various vegetables, bhindi, commonly known as lady's finger, and brinjal are the most common and extensively grown all over India. One of the main constraints of its production is due to diseases caused by several pathogens such as bacterial wilt, Cercospora leaf spot, little leaf in brinjal, YVMB powdery mildew, fusarium wilt in bhindi. The present topic will deals with the symptomatology and management of important diseases of bhindi and brinjal. First of all, diseases of brinjal. Most important disease is the bacterial wilt. It is caused by Ralstonia solanacearum. Now coming to the symptoms of the disease. Wilting, stunting, yellowing of the foliage and finally collapse of the entire plant are the characteristic symptoms of the disease. Lower leaves may droop first before wilting occurs. The vascular system becomes brown. Bacterial ooze comes out from the affected parts. Plants show wilting symptoms at noontime and recover at nights, but will die soon. Next is the mode of spread and survival. The bacterium infects banana, chili, fennel, ginger, potato, radish, tomato, etc. The bacterium is found to be alive and viable for more than 16 months under laboratory conditions and about 10 months on infected plant debris. Presence of root knot nematode, that is Meloidogyna javanica, increases the wilt incidence. Now coming to the management of the disease. Crop rotation with cruciferous vegetables such as cauliflower helps in reducing the disease incidence. Fields should be kept clean and affected plant parts should be collected and burnt. Spray copper fungicide at the rate 0.2% to control the disease. Growing marigold in rotation or as intercrop will suppresses the pathogen in addition to its anti-nematode effect. Growing brassica species and incorporating the plants into soil at flowering stage will reduce disease incidence. Application of organic manures like FOM or poultry manure in every year will increase the beneficial soil microflora and reduce the disease incidence. Use resistant varieties such as Singnath, Islampuri, etc. Next is the biological control. Nursery application. Treat the seeds with talc-based formulation of antagonistic Pseudomonas fluorescens at the rate 10 gram per 100 gram of seeds and soil application of antagonistic Pseudomonas fluorescens 50 gram mixed with 1 kg of soil and incorporated in the nursery bed. Main field treatment. Dip the seedling in Pseudomonas fluorescens or Bacillus subtilis at the rate 10 gram talc formulation per liter of water solution for 20 to 30 minutes just before transplanting. Next disease is the Cercospora leaf spot. It is caused by Cercospora solani, melongeni and Cercospora solani. Symptoms of the disease. The leaf spots are characterized by chlorotic lesions angular to irregular in shape, which later turn grayish-brown with profuse sporulation at the center of the spot. Severely infected leaves drop off prematurely, resulting in reduced fruit yield. Next is the mode of spread and survival. The disease is spread by airborne conidia. Now coming to the management of the disease. Diseases can be managed by growing resistant varieties. Pan Samrad variety is resistant to leaf spot diseases. Spraying 1% body mixture or 2 gram copper oxychloride or 2.5 gram GNAP per liter of water effectively controls leaf spots. Next disease is the Alternaria leaf spot. It is caused by Alternaria melongeni, Alternaria solani. Symptoms of the disease. The two species of Alternaria occurs commonly, causing the characteristic leaf spots with concentric rings. The spots are mostly irregular, 4 to 8 mm in diameter, and may coalesce to cover large areas of the leaf blade. Severely affected leaves may drop off. A. melongeni also infects the fruits, causing large, deep-seated spots. The infected fruits turn yellow and drop off prematurely. Next is the mode of spread and survival. The disease is spread by wind-borne conidia. Now coming to the management of the disease. Spraying 1% body mixture or 2 gram copper oxychloride or 2.5 gram GNAP per liter of water effectively control leaf spots. Next is the little leaf of brinjal. It is caused by phytoplasma. Now coming to the symptoms of the disease. The characteristic symptoms is smalling of the leaves. The petioles are short and the leaves appears to be sticking to the stem. Such leaves are narrow, soft, smooth and yellow. Newly formed leaves are much shorter. The internodes of the stem are also shortened. Next is the mode of spread and survival. The disease is transmitted by leaf hoppers. Coming to the management of the disease. The severity of the disease can be reduced by destruction of affected plants and spraying of insecticides. New crops should be planted only when diseased plants in the field and its neighborhood have been removed. Spraying with insecticides like methyl-dimetone 25 EC or dimethoate 30 EC or malathion 50 EC at the rate 2 ml per liter has been recommended for vector control. Although mycoplasmas are reported to be suppressed by tetracycline, field application of this method has not yet been recommended. Use of resistant varieties such as Chaklasi Doli, Doli 5, Pusa purple cluster. Next important disease is the fruit rot. It is caused by Phomopsis vaccinans. Symptoms of the disease. Affects all above ground plant parts. Spots generally appear first on seedling stems or leaves. Girdle seedling stems and kill the seedlings. Leaf spots are clearly defined, circular up to 1 inch in diameter and brown to grey with a narrow dark brown margin. Fruit spots are much larger. Affected fruit first become soft and watery. but later become black and mummified. Center of the spot become gray and black pycnidia develop. Mode of spread and survival. The fungus survives in the infected plant debris in the soil. It is seed-borne. The spores are spread by rain splashes and through implements and insects. Now coming to the management of the disease. Seeds should be dipped in hot water at 50 degrees centigrade for 30 minutes. Spraying with difolaton at the rate 0.2% or Captan at the rate 0.2% in the nursery and filled at 7 to 10 days interval controls the disease. Deep summer plowing, 3 years crop rotation and collection and destruction of diseased plant debris. Spraying the crop in the field with GNAP at the rate 0.2% or body mixture 0.5% is also effective. Next, coming to the diseases of bhindi. First one is the damping off. It is caused by Pythium species and Rhizoctonia species. Now coming to the symptoms of the disease. Cool, cloudy weather, high humidity, wet soils, compact soils and overcrowding especially favors development of damping off. Damping off kills seedlings before or soon after they emerge. Infection before seedling emergence results in poor germination. If the decay is after seedling emergence, they fall over and die, which is referred to as damp off. The destructiveness of the disease depends on the amount of pathogens in the soil and on environmental conditions. On seedlings, lesions develop on the tender stem near the soil surface. The tissues beneath the lesion become soft, due to which the seedling collapses. Next is the management. Excessive irrigation should be avoided to reduce humidity around the plants. Seed treatment with antagonistic fungal culture of Trichoderma viridi 3 to 4 gram per kg of seed or Thiram 2 to 3 gram per kg of seed and soil drenching with copper oxychloride at the rate 0.2% or Dithan M45 at the rate 0.2% or Babistin 0.1% affords protection against the disease. The field should be regularly inspected for the disease affected seedlings. Such seedlings should be removed and destroyed. Next, coming to the Fusarium wilt. It is caused by Fusarium oxysporum forma specialis vas infectum. Coming to the symptoms of the disease. These wilt pathogen persist in the soil for a very long time. Initially, the plants show temporary wilting symptoms which become permanent and progressive, affecting more vines. The leaves of affected plants show yellowing, lose turgidity and show drooping symptoms. Eventually, the plant dies. In older plants, leaves wilt suddenly and vascular bundles in the collar region become yellow or brown. The fungus invades the root system and colonizes the vascular system. In doing so, water movement is blocked and toxins from the fungus alter normal cell function. Cutting the base of the stem reveals a dark woody portion. Now coming to the management of the disease. Continuous cultivation of bhindi in the same field should be avoided. In case of fields severely infected by the wilt pathogen, practicing of long crop rotations is useful in reducing the pathogen population. 3 sprays of Carathan at the rate 6 gram per 10 liter of water or Babistin at the rate 1 gram per liter of water immediately on appearance of initial symptoms at 5 to 6 days interval checks the spread of the disease. Leaves of fully grown plants should be thoroughly drenched during spraying. Next disease is the powdery mildew. It is caused by Ericephale cicoracearum. Symptoms of the disease. The disease symptoms appears as subtle, small, round, whitish spots on leaves and sometimes on stems. The spots enlarge and coalesce rapidly, and a white mass resembling talcum powder becomes evident on the upper surface of the older leaves and other plant parts. A large part of the talc-like powder on the leaf surface is composed of spores. These spores are easily blown by winds to nearby susceptible plants. Heavily infected leaves become yellow, then become dry and brown. Extensive premature defoliation of the older leaves can ensure that the disease is not controlled. Coming to the management of the disease. Healthy, vigorous leaves and stems are less prone to infection. Plants under nutritional stress in most cases will develop powdery mildew much sooner than plants grown under a good nutritional program. Hence, the plants should be well manured and application of fertilizers should be done on the basis of standard recommendation. Application of wettable sulfur at the rate 0.2% or Babistin at the rate 0.1% 2 to 3 times at an interval of 1 week effectively control the disease. Next disease is the Cercospora leaf spot. It is caused by Cercospora abelmosi and Cercospora malayensis. Symptoms of the disease. Cercospora malayensis causes brown irregular spots and Cercospora abelmosi causes sooty black angular spots. The affected leaves roll, wilt and fall. The disease causes severe defoliation during humid seasons. Next is the management of the disease. Removal and destruction of diseased plant materials helps to check the spread of the disease. The disease is effectively controlled by spraying with copper oxychloride at the rate 0.3% or Zineb at the rate 0.2% starting from about a month after sowing and repeating at fortnightly intervals depending upon the severity of the disease incidence. Next is the yellow vein mosaic. It is caused by yellow vein mosaic virus. Coming to the symptoms of the disease. This is most important and destructive viral disease in Bhindi. The disease is transmitted by whitefly. The characteristic symptoms of the disease are a homogeneous interwoven network of yellow veins enclosing islands of green tissues. Initially infected leaves exhibit only yellow colored veins, but in the later stages, the entire leaf turns completely yellow. In extreme cases, the infected leaf becomes totally light yellow or cream colored and there is no trace of green color. Plants infected in the early stages remain stunted. The fruits of the infected plants exhibit pale yellow color, deformed, small and tough in texture. Next is the management of the disease. Removal and destruction of virus affected plants and planting of disease-free material. Resistant hybrids such as Makhmali, Tulsi, Anupama 1 and Sun 40 should be grown during Kharif season. Soil application of carbofuran at the rate 1 kg AI per hectare at the time of sowing and 4 to 5 foliar sprays of dimethoate at the rate 0.05% or metacistox at the rate 0.02% or Novacron at the rate 0.05% at an interval of 10 days effectively controls the whitefly population. So this is all about the diseases. Now coming to the conclusion of the topic. Brinjal and bhindi both are consumed as tender vegetables, due to which they are more prone to disease attack. There are several pathogens which hamper its productions, among them bacterial wilt, Cercospora leaf spot, little leaf and fruit rot are important diseases of brinjal, and in bhindi they are damping off, fusarium wilt, powdery mildew and yellow vein mosaic virus. Management of the diseases depends upon the proper diagnosis, timely application of control measures. Among the management practices, best one is the use of resistant varieties if available for the diseases.